Welcome, curious minds, to another deep dive. Today, we're plunging into a world where surgical precision isn't just a goal, it's an absolute necessity for saving lives. Imagine, if you will, a surgeon performing an incredibly delicate procedure deep inside the human heart. They're trying to precisely target microscopic, faulty electrical pathways, but they're working almost blind, relying heavily on feel, or maybe even worse, without that crucial tactile feedback we often take for granted. That, surprisingly, has long been the reality in many sensitive heart surgeries. Okay, let's unpack this. We're diving deep into a fascinating piece of engineering and medical innovation, a ring-shaped, MEMS-based force sensor. Now, for the technical part, and we'll explain it all, I promise it's piezoresistive, which just means it cleverly changes its electrical resistance when it's squeezed or stretched, by mechanical stress. This tiny marvel is specifically designed for cardiac ablation catheters. It's a mouthful, I know, but trust me, the implications for making heart surgery safer, more effective, they're absolutely immense.
Yeah, and what's truly fascinating here is how a technological advancement, something developed on a microscopic scale, can address such a macroscopic and, well, critical challenge in patient care. We're talking about a tiny sensor, you can barely see it, that has the potential to make a monumental difference in treating one of the most common and dangerous heart rhythm disorders. This innovation is really about bringing an unprecedented level of real-time control and feedback right there to the operating table. It could fundamentally change how these procedures are done.
And to guide us through this intricate landscape of microelectronics and cardiac health, we've got a detailed research paper. It's from the IE Sensors Journal. This paper meticulously walks us through how they designed it, how they built it, and how they thoroughly tested this groundbreaking device. Our mission for this deep dive: to extract the most important nuggets of knowledge, understand the why and how behind this innovation, and really explore what it means for the future of cardiac care. We want to give you that shortcut to being truly well-informed. So if you've ever found yourself wondering how technology constantly pushes the boundaries in medicine, or maybe if you're just insanely curious about those aha moments in engineering, the ones that fundamentally change how we approach complex problems, well, you are absolutely in for a treat. This is where cutting-edge science truly meets the very heart of human health. Let's pivot now to the sheer scale of the problem this sensor is designed to address. This isn't just some niche medical issue. Heart rhythm disorders, medically known as cardiac arrhythmias, or CA, they're a significant and growing part of non-communicable diseases globally. We're talking about a massive health burden that touches millions of lives across every continent. It affects people from all walks of life.
To give you a real sense of the global impact, the sources tell us some truly sobering numbers. I mean, a staggering 26 million people worldwide currently suffer from heart failure, and that condition is often complicated by these underlying arrhythmias. And if we look at specific regions, in India alone, approximately 0.05 million people, that's 50,000, receive interventional devices annually just to manage these heart rhythm issues. Among the various types, atrial fibrillation, or AF, stands out. It's the most common rhythm disorder across the globe, and its impact isn't just about discomfort. It was responsible for a shocking 42.8% rise in global deaths between 2006 and 2016.
Wow, nearly 43% increase.
Exactly, and the projections for the future, they paint an even more dramatic picture. The occurrence of AF is expected to increase to between 6 and 12 million in the USA by 2050, 17.9 million in Europe by 2060, and a truly immense 72 million in Asia by 2050.
72 million.
Yeah. This isn't just a medical statistic. It's a rapidly expanding global health crisis. It profoundly impacts quality of life and, well, it demands urgent and innovative solutions. Imagine the stress, the fatigue, the constant worry that millions face every single day because of this.
Absolutely. And with such a widespread and serious issue, medical science is naturally focused intensely on developing effective solutions. One of the most significant advancements, and it's minimally invasive, has been catheter ablation. This procedure uses focused radiofrequency, or RF energy, to treat these arrhythmias. The goal is to basically eradicate faulty electrical pathways in heart tissues by ablating, which means precisely burning specific targeted areas. This creates a controlled lesion as a small localized area of tissue damage leading to tissue necrosis, which is the intentional, deactivation of those problematic cells. Think of it like finding a faulty wire in a really complex electrical circuit and carefully surgically deactivating just that segment to restore proper function.
Right. But this raises an important question. If it's so effective and minimally invasive, what's the catch? What's the challenge? You see, despite its advanced nature, the procedure's success hinges entirely on effective lesion formation, meaning you have to create just the right amount of damage in exactly the right place to silence those rogue electrical signals. And that, in turn, critically depends on a factor that's surprisingly difficult to gauge in real time: the contact force, the force between the catheter tip and the delicate, constantly moving heart tissue. Without extremely precise control over this force, even the most skilled surgeon can face significant hurdles.
You've really pinpointed the heart of the matter there. This isn't just about applying some force. It's about hitting an incredibly precise sweet spot. Imagine trying to gently poke something incredibly delicate with a long, flexible stick, but you can't actually feel how hard you're pushing. You're working almost entirely by sight, from a distance. That's essentially the challenge operators face during these procedures. There's a Goldilocks zone for quality ablation, a force that's neither too much nor too little. It's incredibly narrow. Unforgiving, really. We're talking about an optimum force range of just 0.2 to zero point 0.4 newtons. To put that into perspective, that's roughly the weight of, maybe a couple of standard paper clips. Applied with pinpoint accuracy inside a beating organ, the margin for error here is almost non-existent. So what happens historically when surgeons miss that tiny window without precise feedback?
Well, the stakes are incredibly high for the patient. Let's consider the consequences of getting it wrong. If the contact force is insufficient, meaning below that optimal zero point 0.2 newtons, it leads directly to poor or incomplete ablation. Just doesn't work properly. The faulty electrical pathways aren't fully eliminated, and the patient faces a much higher chance of atrial fibrillation recurrence.
Meaning they might have to go through it all again.
Exactly. Often means they have to undergo the discomfort and risks of the procedure again, or worse, just continue to suffer from the debilitating symptoms. On the flip side, if the force is too high, say above zero point 0.4 newtons, it can cause the heart tissue to kind of enfold over the catheter tip. Now, that might sound benign, but it severely reduces the effectiveness of the radiofrequency current delivery, and it prevents proper electrode cooling. Both of those are essential for a clean, effective, and safe ablation. And here's the truly critical part, something observed in stark porcine, that's pig in vivo experiments. Forces above 0.77 newtons can actually perforate heart tissues.
Perforate, like poke a hole through it.
Precisely. A hole in the heart wall. That's a catastrophic outcome, potentially leading to life-threatening complications like cardiac tamponade. So as you can see, it's a razor-thin line between healing and causing further severe harm. The need for precise, real-time force measurement isn't a luxury in this procedure. It's an absolute necessity for patient safety and procedural success. Historically, surgeons relied on their experience, their feel, and fluoroscopy, that's like a live x-ray which shows catheter position, but not the actual contact force. It was a skilled art, absolutely, but one inherently limited by a lack of objective, real-time data.
So when we talk about what this all means for the surgeon and the medical team performing these intricate procedures, it paints a picture of immense challenge. The existing procedure, despite its life-saving potential, grapples with significant hurdles. We're talking about the immense difficulty of navigating a flexible catheter to the precise target location within the complex moving anatomy of the heart. It's not easy. There's a profound lack of adequate visual information that would allow them to truly see the contact force they're applying. And most critically, as you said, insufficient force feedback, that tactile sense that tells you exactly how hard you're pressing. On top of all these technical anatomical challenges, there's the very human element: operational fatigue, stress, experienced by the medical professionals while working with such small, specialized tools in high-stakes situations. It's a recipe for potential complications, leading to longer procedure times and increased risk for the patient.
Precisely. And despite significant advancements in catheter technology over the years, including the integration of early forms of force sensing like strain gauges and fiber optic technologies, the sources we looked at indicate that a truly MEMS-based force sensor, one fully compatible with the specific requirements of an ablation catheter, simply hadn't been reported. And this isn't just about measuring force, it's about integrating it seamlessly into a device that also needs to allow for saline irrigation through the catheter tip.
The irrigation. Why is that so critical?
That irrigation flow is absolutely vital during ablation. It constantly cools the tip and flushes the area. This prevents the formation of char-burnt tissue and dangerous blood clots, or thrombus, which can severely compromise the ablation's effectiveness and, critically, the patient's safety. A clot breaking off could cause a stroke.
Okay, so irrigation is non-negotiable.
Absolutely. So this gap, the lack of a suitable MEMS sensor that didn't interfere with irrigation, meant that operators were often, as you said, flying blind, especially regarding precise real-time contact force. They were relying heavily on their highly trained hands' years of experience, which, while impressive, couldn't provide the consistent, objective data needed to stay reliably within that critical Goldilocks zone we discussed. Previous attempts at force sensors often added too much bulk, or they interfered with other critical functions like that essential irrigation, making them impractical for this specific application.
Okay, so this is where the magic of miniaturization truly shines, right? Delivering a solution to that critical problem. We keep talking about MEMS, or micro-electromechanical systems. For those unfamiliar, can you give us a quick definition? These are miniature devices that ingeniously combine electrical and mechanical components, all on a microscopic scale. Think of it as shrinking an entire intricate machine, maybe with tiny moving parts, incredibly sensitive sensors, clever actuators down onto a silicon chip, sometimes smaller than a grain of sand. It's truly a marvel of modern engineering, allowing us to interact with the world at a scale previously unimaginable.
And this is absolutely critical because MEMS technology enables the integration of incredibly sensitive and precise sensors into extremely small spaces. As we've highlighted, that's exactly what a catheter tip requires, a tiny yet robust and unobtrusive sensing element. It needs to fit within the existing dimensions of an ablation catheter without impeding its other crucial functions, like irrigation or navigation. It represents a significant leap beyond traditional larger strain gauges, not just in terms of sheer scale, but also in its potential for seamless integrated operation. It really opens up entirely new possibilities for real-time feedback that were previously unattainable. This isn't just about making things smaller, it's about enabling functionality that just wasn't possible before at this scale.
And the paper's core contribution, the big aha moment here, is precisely this. A piezoresistive MEMS sensor specifically designed to measure catheter tip contact force in real time. This isn't just about a post-procedure analysis or some general indication of pressure. It's about providing immediate, accurate, and continuous feedback during the procedure. It gives the surgeon an unprecedented level of control, almost like giving them a new microscopic sense of touch right there inside the heart.
Exactly. And to elaborate on that piezoresistive part, these are remarkable materials. They fundamentally change their electrical resistance when they're subjected to mechanical stress. Imagine a tiny, specialized electrical pathway. When you push it or pull it, it alters how easily electricity flows through it. This change in electrical resistance is precisely what the sensor measures. And with careful calibration, it can be directly and precisely translated into a quantified measurement of the applied force. The inherent beauty of piezoresistance is its high sensitivity and its, well, ideal compatibility with silicon-based MEMS structures. This makes it perfect for integrating into such a small, delicate, and yet demanding medical application where sensitivity and miniaturization are paramount. It effectively allows the catheter tip to feel the pressure and convert that feeling into an electrical signal the surgeon can see and react to instantly.
The actual architecture of this sensor is remarkably elegant in its simplicity, yet incredibly sophisticated in its execution. You described it as like a tiny donut, or maybe a very small inner tube, right? With 4 identical bridges connecting its central inner ring to its outer ring. It's a very clever mechanical design, but the true genius lies in what's on those bridges. Can you walk us through those crucial sensing elements?
Indeed, these 4 bridges are absolutely key. They act as the primary load-bearing and sensing components, and each bridge is incredibly small. To give you a true sense of scale, we're talking about structures roughly 500 micrometers long, 200 wide, and only 30 thick. That's incredibly thin.
Thinner than a hair.
Much thinner. And importantly, embedded within each of these tiny bridges are 2 even smaller bar-shaped P-type piezoresistors. Each is about 40 by 10 micrometers. Now, these aren't just any resistors. They are specifically boron-doped silicon and meticulously oriented in the silicon's 110 crystal direction. That specific orientation is known to maximize their sensitivity to mechanical stress. It makes them feel the strain more effectively. This particular design, with 2 piezoresistors per bridge, is a notable departure from many previous sensor configurations that often use just a single piezoresistor, or maybe different layouts for normal versus shear forces. This dual resistor setup on each bridge, combined with the 4-bridge ring design, not only makes the sensor highly sensitive to normal forces, the direct push, but also inherently allows for the potential measurement of shear forces. Those are the forces acting parallel to the surface, like sliding forces. That's for future work, though. The overall change in resistance from all 8 piezoresistors across the 4 bridges can be measured, providing a comprehensive picture of the forces acting on the tip.
So it's not just about a simple push, but potentially a much richer, multidimensional understanding of how the catheter is interacting with the tissue. It's fascinating. And to connect this back to the surgical need, the entire sensor is meticulously optimized to measure forces specifically between 0 and 0.8 newtons. Why is that specific range so important again?
That 0 to zero point 0.8 Newton range is precisely the critical window for cardiac ablation procedures. As we discussed earlier, falling below zero point 0.2 newtons means ineffective ablation. The lesion just isn't good enough. Exceeding zero point 0.4 newtons can lead to that tissue unfolding and reduced efficacy. And critically, going above that danger threshold of around 0.77 newtons risks perforating the heart tissue itself. By optimizing the sensor for this specific narrow range, the researchers have ensured that it provides the most precise and actionable data exactly where it's needed most. It allows operators to stay safely and effectively within that crucial Goldilocks zone we mentioned, avoiding both under and over ablation. It's about providing the right information at the right time to make the right decision.
And the innovation isn't just in the sensor itself, is it? It's also about how it's fully and intelligently integrated into the catheter. The researchers didn't just design a standalone sensor, they designed a customized ablation electrode tip to perfectly house it. This tip has a diameter of 3.5 millimeters and a height of 4 millimeters. You mentioned these dimensions are consistent with existing clinically used catheters.
Exactly. That's crucial for adoption. It ensures it can slot seamlessly into current medical practice without requiring a complete overhaul of the equipment doctors are used to.
That's a really crucial detail. It's not about reinventing the entire catheter, but enhancing a key component.
Precisely. It's about a targeted, intelligent enhancement. And to elaborate on the design features of this customized tip, it's ingeniously engineered with what's called a force transfer structure, or FTS. This FTS itself is like a small cylinder, same dimensions as the tip, with a hole through it for the electrode. On its bottom surface, it features 4 precisely crafted pillars, each tiny 200 micrometers by 200 micrometers. These pillars are absolutely critical because they are meticulously aligned to the center of the sensor bridges we just discussed.
So they make direct contact.
Exactly. These pillars, which are slightly rounded or filleted at their edges to ensure precise positioning, ensure that the forces exerted by the catheter tip onto the heart tissue are directly and accurately transferred to the sensor's bridges. It's like having a perfectly engineered microscopic pressure distributor, ensuring the signal from the tissue gets cleanly to the sensor.
So essentially, the FTS acts like a tiny, incredibly precise scaffold within the catheter tip. It's designed to funnel that subtle pressure from the heart tissue through those miniature pillars directly onto the sensor's bridges. It's a clever way to ensure no signal is lost and every bit of force is accurately captured. Very smart.
Exactly. But there's another crucial element we touched on. The tip also includes that stainless steel pipe for saline irrigation. It has an outer diameter of 0.9 millimeters and an inner diameter of 0.6 millimeters. And as we said, this irrigation flow is absolutely vital during ablation. It constantly bathes the electrode tip in saline solution. Why is this so important? Firstly, it prevents char and thrombus formation, which can compromise the procedure and create dangerous debris. Secondly, it ensures maximum power transfer from the electrode to the tissue by cooling the tip. This prevents overheating at the very surface and allows for deeper, more effective lesion formation without causing superficial damage. Without it, the ablation would be far less effective and significantly riskier. The sensor itself is cleverly sandwiched between the catheter's shaft coupler and this force transfer structure. This provides both robust mechanical support and the necessary electrical connections. It's an entire, sophisticated system designed for optimal and safe performance.
And to add another layer of clever design, you mentioned the researchers also thoughtfully included 2 additional piezo resistors, but these are on the outer ring structure. You said they aren't for direct force sensing in the same way, but act as temperature compensation sensors within the signal conditioning circuits. That sounds like a very smart detail. Why would temperature fluctuations be a concern, and how do these compensation sensors address that?
That's a really perceptive question. Temperature is a significant factor because the electrical resistance of piezoresistors, like many electronic components, can change with temperature. It's just a physical property. So if the temperature around the sensor fluctuates during a procedure, which is certainly possible inside the body, or even due to the RF energy itself, those thermal changes could subtly affect the resistance readings. It would make it difficult to differentiate between a change in resistance due to applied force and a change due to temperature. It would essentially introduce noise or error into the force measurement.
I see. So a temperature spike could look like a force spike.
Potentially, yes. Or it could mask a real change in force. By including these dedicated temperature compensation resistors, which are placed on the outer ring where they experience the same thermal environment but don't experience the direct mechanical stress from the bridges, the system can measure the resistance change caused solely by temperature. Then, the signal conditioning circuit can effectively subtract that thermal effect out from the signal coming from the main force-sensing resistors. This ensures that the final force readings are pure, accurate, and truly reflect the contact force, making the sensor much more robust and reliable in a dynamic environment like the human body. It really speaks to the holistic, careful approach taken in the design.
Okay, so they had the blueprint. They knew exactly what they wanted this tiny sensor to do. But designing it on paper is one thing. Actually building something this intricate at a microscopic scale, that's where the real engineering wizardry comes in. How do you even begin to craft something smaller than a grain of sand with such precision before committing to physical fabrication, which I imagine is incredibly costly and time-consuming at this scale?
Right. Absolutely.
The team behind this innovation rigorously modeled their design. They use powerful software called COMSOL multiphysicses for extensive finite element modeling, or FEM simulations. Think of this as creating a virtual prototype, right? A digital dress rehearsal to test everything before they even started building.
Exactly right. These simulations were incredibly thorough and serve three critical goals. First, they aim to analyze the precise structural behavior of the sensor itself, how it would deform, where the stresses would concentrate under various loads. Second, they were used to pinpoint the absolute optimal position for those piezo resistors on the tiny bridges. You want them in the areas of maximum stress for the highest possible sensitivity, essentially, where they could feel the most strain. And third, and perhaps most complex, they meticulously simulated how the force is actually transferred to the sensor once it's fully integrated into that customized catheter tip with the FTS. To do this, they applied simulated forces ranging from 0 to 0.5 newtons to the virtual bridges, identifying those high-stress locations, and then iteratively optimized the piezo resistor dimensions for even better sensitivity. It's a meticulous process of virtual refinement. It saves immense time and resources compared to just building and testing physical prototypes blindly.
And the simulation showed something really important and reassuring, didn't they? You mentioned that when force is applied to the catheter tip, it is equally distributed among those four bridges. That seems absolutely key for accurate measurement because it means the sensor isn't just detecting force at one point. It's getting a balanced, comprehensive reading across its entire structure. That should lead to much more reliable data for the surgeon.
Precisely. And to put some numbers on those insights, the simulations demonstrated what's called a normalized sensitivity of 0.076 NA when the force was applied directly on the sensor bridges in the simulation. Now, normalized sensitivity essentially gives us a standard way to measure how much the sensor's electrical signal changes for every unit of force applied relative to its base resistance and the voltage used. When the force was applied to the fully integrated catheter tip simulation, the normalized sensitivity was slightly lower, around 0.018 NA.
Lower. Is that bad?
Not at all, it's expected. This might sound like a drop, but it actually perfectly illustrates the intended force distribution. The resistance change in the integrated tip's piezo resistors was approximately 4 times lower than when the force was applied directly on the bridges. This isn't a design flaw. It confirms that the force is indeed being effectively distributed and transferred through that force transfer structure to all 4 bridges, which is absolutely essential for the design's effectiveness in a realistic integrated setup. It showed that their clever FTS design worked exactly as intended, even before a single physical sensor was produced. That must have built immense confidence moving into the fabrication stage.
Okay, so the simulation's checked out. Now for the really hard part. Creating something this small, this precise, with such complex internal structures. It's not just design, it's a micro-manufacturing marvel. You described it as a complex, multi-step process, often carried out in ultra-clean rooms, demanding incredible precision at every single stage. The sensor itself, with its 3.5 millimeter diameter, was built on a 3-inch silicone wafer. Can you give us a sense of what that fabrication process looks like? How do you even begin to craft something so tiny and intricate? It sounds like science fiction.
It almost is. Bringing the sensor to life is truly a masterclass in micromanufacturing. It requires an almost unbelievable level of precision, often in environments cleaner than a surgical operating room to avoid even microscopic dust contamination. Imagine building a tiny, intricate sculpture layer by layer, starting with a perfectly smooth silicone wafer, which is incredibly thin, just 360 nanometers thick in this case, much thinner than I said before. That was wafer thickness.
Okay, nanometers, wow.
Yeah, incredibly thin. They began by carefully growing a thin, insulating layer of silicon dioxide, SiO2, essentially a high-quality glass layer on this wafer using thermal oxidation, about 500 nanometers thick. Then, using the first of 6 specialized masks or stencils and a process called photolithography, using light to pattern materials, they meticulously patterned this oxide layer, defining where the first sensor components would go. This is followed by a critical step, boron diffusion. This is where boron atoms are precisely introduced or doped into the silicon in those patterned areas, creating the p-type piezo-resistive elements, those tiny sensitive regions that will change resistance when stressed.
So that's how they make the actual sensing part.
That's step 1 for the sensing part. After removing some temporary glass layers, they deposit another layer of SiO2, pattern it with mask #2, and then diffuse highly doped boron to create low-resistance contacts between the piezoresistors. Good electrical connection is key. Then, they insulate the piezoresistors again with another layer of SiO2, pattern that with mask #3 to open windows for contact pads. Then they deposit and pattern aluminum using mask #4. This aluminum forms the actual electrodes, the metal pathways that will connect the tiny sensor to the outside world, to the wires, and eventually the monitoring equipment the surgeon will see.
Okay, so that's building up the layers. What about creating the bridges and releasing the sensor?
Right. Now comes the etching, the carving part. They pattern the top side of the wafer again using mask #5. Then they etch through the SiO2 layer and use a powerful technique called deep reactive ion etching, or DRD, to etch 50 micrometer deep trenches into the silicon itself. This starts defining the sensor structure. And then, in a truly remarkable step, they flip the wafer over and pattern the backside using mask #6. They etch the SiO2 on the back and then use DRA again, this time etching away about 330 micrometers of silicon from behind. This final deep etch from the back is what carefully carves out and undercuts the structure, creating those tiny suspended bridges we discussed earlier and ultimately releasing the entire ring-shaped force sensor from the main silicon wafer.
So they etch almost all the way through from the back to free it up.
Exactly. It's a painstaking process requiring precision down to just a few micrometers or even nanometers at every single step. Think of it like a sculptor working with chemicals, plasma, and light instead of a chisel to create something incredibly small and functional. The final product is this intricate, fully realized force sensor, truly a marvel of microfabrication, ready for its very big job.
Okay, so once you've gone through all that meticulous design, simulation, and incredibly complex fabrication, the sensor needs to be rigorously tested. You can't just assume it works. You have to prove it step by tiny step. First up, its basic electrical properties. Does it conduct electricity as expected and predictably?
Absolutely. For the electrical characterization, they used a probe station which has tiny needles to make contact with the sensor's pads and a sophisticated source measurement unit, a Keithley 20636B. They applied a sweep voltage from 0 to 10 volts across the individual piezoresistors and precisely measured the current flowing through them. This initial testing revealed a base resistance of approximately 1276 ohms for the piezoresistors, with some variation, 240A, which is normal in fabrication. But critically, it showed a beautifully linear IV current voltage characteristic. That linearity is exactly what you want for a sensor like this. It indicates predictable and reliable electrical behavior across its operating range. It means that if you double the voltage, you essentially double the current, which is ideal for accurately translating resistance changes into force measurements.
Okay, so electrically, it behaves well. But beyond just electricity, you need to see if it looks right microscopically. We're talking about components smaller than a human hair, so when you could design something perfectly on a computer, microfabrication has its own challenges, as you mentioned. Physical verification seems absolutely essential to ensure the physical dimensions match the blueprint.
Definitely. For the optical characterization, they used a non-contact 3D optical profiler, specifically a Taylor-Hobson Precision TaliSurf CCI. This is a fancy microscope that can map the surface in 3 dimensions with incredible accuracy. It allowed them to measure the physical dimensions of the fabricated sensor structures, the bridges, the rings. It largely confirmed the intended dimensions. Bridge width around 222 m, a bit wider than designed, likely due to etching effects, thickness around 40 m, and the overall ring thickness around 330 m. Interestingly, they did note some slight variations, like the inner ring being slightly narrower, 290 m, and the outer ring slightly wider, 340 m, than designed. These minor deviations, just a few micrometers here and there, were attributed to using a single-side polished wafer, meaning the back wasn't perfectly smooth, and inherent, unavoidable variability in that deep reactive ion etching DRE process we discussed.
The etching again.
Yeah. It's a fantastic insight into the real-world challenges of even high-precision manufacturing. Even at this tiny scale, absolute perfection is elusive, but the key is that the design must be robust enough to accommodate these minute, real-world variations without compromising overall performance. It shows the incredible precision engineers are working with and the tolerances they have to manage.
Right. So next, the sensor needed to be integrated into a functional assembly for mechanical testing. How did they do that? It was carefully die bonded, essentially glued with a specialized epoxy onto a printed circuit board, or PCB, and then tiny electrical connections were made by wire bonding with thin aluminum wires, like microscopic soldering. The PCB provides critical mechanical support, acting as the sturdy backbone for the delicate sensor. But how did they apply force to something so small and get accurate readings? That sounds tricky.
It is tricky. The mechanical characterization involved a sophisticated two-pronged approach to thoroughly test the sensor's performance under various loads. First, they utilized a Heisatron nanoindenter. This is an extremely precise instrument, equipped with a very sharp diamond microneedle, almost like a microscopic record player needle, but much harder. It was used for initial tests of the sensor's resolution, how small a force change it could detect, and its response times. They applied loads ranging from a very light 0.1 newton up to zero point 0.4 newtons in precise 0.1 newton steps. They incorporated careful 5-second loading and unloading periods, along with a 10-second hold time at peak load. This careful timing wasn't just for accuracy, but also to prevent potential damage to the delicate microneedle itself. The results were quite impressive. The sensor showed average rise in recovery times, how fast it responded to force changes of around 1.8 seconds. That speed is absolutely crucial for real-time feedback in a dynamic surgical environment where conditions can change rapidly and the surgeon needs immediate data.
1.8 seconds. That sounds pretty fast for real-time feedback.
Okay.
What was the second approach?
Second, for testing the sensor across its full working range, up to 0.8 N potential, though tested up to 0.4 N per bridge here, and under more controlled conditions, an in-house automated test setup was developed. This was cleverly automated using LabVIEW software. This setup involved indenting individual bridges of the fabricated sensor using a very fine 100 micrometer diameter pin, much larger than the nano indenter tip, allowing for higher forces. This pin was connected to a commercial load cell, a highly accurate force sensor itself, a Honeywell Model 11 with a 250-gram capacity via a customized holder or cullet. This allowed for precise and measurable force application. The load cell, in turn, was attached to a micromanipulator, providing extremely fine, computer-controlled movement in tiny increments. And here's where it gets truly clever. They used a highly innovative shadowing technique with an LED light source and a high-resolution camera to precisely position that tiny indenter pin on the exact midpoint of each bridge.
Shadowing. How does that work?
Imagine shining a bright, focused light from one side, casting a sharp shadow of the indenter pin and the sensor bridge onto the camera sensor. By analyzing the shadow's position relative to the bridge image, the system could guide the micromanipulator with incredible accuracy to place the pin right in the center. It's a non-contact alignment method, very ingenious for this scale.
That is incredible, using light and shadow to achieve such microscopic precision. So the testing protocol itself was incredibly detailed, I imagine. The micromanipulator displaced the pin in very small, precise 5-micrometer steps, gradually loading each bridge up to 0.4 newtons, that's about 40 grams of force per bridge, and then carefully unloading it. Data was acquired continuously, averaged over 50 readings at each step to ensure accuracy, and saved. They even incorporated a 30-second stabilization time before recording data in each step to minimize any potential measurement errors from environmental factors or instrument settling. That level of rigor really speaks volumes about the quality of the research and their desire for unflinching accuracy.
Absolutely. And the results from this exhaustive testing were truly impressive and, importantly, closely validated the earlier FEM simulations. The P-series sisters demonstrated remarkable linearity of 99.5% for forces applied up to 0.3 newtons on individual bridges. This means the sensor's electrical output was almost perfectly proportional to the applied force within that critical range, extremely predictable. Beyond 0.3 N, some slight nonlinearity was observed, which is common in such materials as they begin to experience higher stresses, but still very usable. Importantly, the maximum hysteresis sets a measure of how well the sensor returns to its original zero reading after a load is removed. Essentially, its memory effect was only 5%. Low hysteresis is crucial for accurate readings during dynamic loading and unloading.
So it doesn't get stuck easily.
Exactly. And the sensitivity, they measured an average resistance change of 108 ohms per newton of applied force. Expressed as normalized sensitivity, that's 0.086856 N1 and 0.00124 Nn. This value is very close to the simulated value for direct bridge loading, 0.076 Nf, further validating both the fabrication and the simulation model. These numbers confirm that this is a highly reliable and responsive sensor capable of providing the nuanced feedback required for cardiac ablation. It makes it a robust and trustworthy tool for surgeons.
That's great validation. Now, interestingly, the researchers also noticed a specific detail. One of the bridges, which they labeled Bridge 1, showed slightly higher sensitivity than the others. This was attributed to it being optically characterized as marginally thinner, measuring around 36 micrometers, compared to the average 40 micrometers for the others. This illustrates how even tiny fabrication variations, down to just a few micrometers, can subtly impact performance. What does this tell us about the real-world challenges of microengineering?
It's a fantastic, practical example of the realities of microfabrication. Even with the most advanced techniques, achieving perfect uniformity across an entire wafer of hundreds or thousands of these tiny sensors is incredibly difficult. A variation of just 4 micrometers, which is minuscule to us, can indeed have a measurable impact on the mechanical properties and thus the sensitivity of an individual component like a bridge. However, what's significant here is that the overall design of the sensor, with its 4 bridges and 8 piezo resistors, is inherently robust. It's designed to provide an accurate aggregate measurement, typically using a Wheatstone bridge configuration that averages out or compensates for minor variations between individual elements. So while one bridge might be slightly more sensitive, the combined data processed through the right circuitry still provides a highly reliable overall force reading. It speaks to the engineering acumen of not just designing a perfect sensor on paper, but designing a system that can function effectively despite those inevitable real-world imperfections.
Right, designing for resilience. And building on that, another fascinating insight came from the testing on a different bridge, Bridge 2. They noted that a slight positioning error during indentation, maybe the shadowing technique, wasn't perfect, that one time could lead to a torsional load, essentially a twisting force, rather than a pure compressive push straight down. This caused one piezoresistor in that bridge to experience an increase in stress, while the other experienced a decrease, impacting the individual readings from those two resistors. How does the overall system cope with such variability, or even these unintended twisting or sideways forces?
That's a great observation, and it highlights the complexity, but also the potential of multi-axis force sensing. As you described, while a purely compressive force, normal force, would cause both piezoresistors in a bridge to change resistance in the same direction, either both increase or both decrease depending on placement and doping, a torsional or shear component would cause them to change in opposite directions, or at least with different magnitudes, because one side of the bridge is being stretched while the other is compressed, or vice versa. The beauty of having multiple piezoresistors, 2 per bridge, and multiple bridges, 4 total, is that the system can be configured through clever wiring, like multiple Wheatstone bridges, to not only measure the primary normal force by summing signals, but also potentially detect and even quantify these shear or torsional components by analyzing the differential resistance changes between resistors on the same bridge or between bridges.
So the difference tells you about twisting.
Exactly. For the immediate purpose of this paper, accurately measuring normal force, even with that slight positioning error and the resulting mixed load on bridge 2, the sensor still provided data within a functional range when considering the overall system response. But this inherent capability, where different types of force affect the resistors differently, beautifully lays the groundwork for future development towards triaxial force sensing, measuring forces in all 3 dimensions, up, down, side to side, front to back. That would provide an even richer, more complete data set for the surgeon about how the catheter is interacting with the tissue. It means the sensor design is not only resilient, but also adaptable for future enhancements.
Okay, so simulation, fabrication, benchtop testing, it all looks great. But the ultimate test for any medical device, especially one designed for such a critical application inside the heart, isn't just in the sterile confines of the lab, it's how it performs on actual biological tissue.
Yeah.
This is where the rubber meets the road, or maybe where the sensor meets the myocardium. This is the crucial step in bridging the gap between theoretical performance and real-world clinical viability.
Precisely. You absolutely have to test it in conditions that mimic the clinical scenario as closely as possible. So, to bridge that critical gap, a complete catheter tip assembly was developed. This included integrating the fabricated MEMS sensor within a customized catheter tip made of SS316 stainless steel, a common medical-grade steel. Interestingly, this tip was produced using metal 3D printing, which allows for complex geometries. This entire assembly, sensor inside the metal tip, was then carefully mounted onto that same micromanipulator setup from the benchtop tests. This allowed for precise uniaxial compression tests, straight pushing to be carried out on excised porcine heart tissues.
Porcine meaning pig hearts. Why use those?
Yes, pig hearts. They are widely used in cardiac research because they are anatomically and mechanically very similar to human hearts in terms of size, structure, and tissue properties. This makes them an excellent and ethically responsible model for preclinical testing before ever considering human trials.
And it's important to note the ethical considerations here. You mentioned the paper states that all animal protocols for this research were meticulously reviewed and approved by the Institute Ethical Committee of the Indian Institute of Science, ISEC, in Bangalore, India. This ensures that the research was conducted responsibly and ethically, adhering to strict guidelines for animal welfare.
Absolutely essential. For the tests themselves, the porcine hearts were obtained fresh from a local slaughterhouse. They were carefully preserved in a physiological saline solution, kept cool at around 4 degrees Celsius during transportation to the lab. Testing was then performed within 2 hours of retrieval to ensure the tissue properties were as close to living tissue as possible, conducted at a controlled room temperature of 20 degrees Celsius. Tissue blocks, approximately 15 millimeters square and 5 millimeters thick, were meticulously prepared from different regions of the heart, specifically samples from both the left and right atria, the upper chambers, and the left and right ventricles, the main pumping chambers. Each tissue block was indented 3 times at different locations, about 5 millimeters apart, applying forces up to 0.4 newtons using the sensor-integrated catheter tip. This comprehensive approach allowed them to thoroughly assess the sensor's interaction with different regions and properties of the heart, which is crucial because, as we'll see, not all heart tissue behaves the same way.
Right. And what's fascinating here is not just the sensor's performance during these tests, but also what they learned or confirmed about the heart tissue itself. It's not just a uniform block of material, it's a dynamic biological structure. What did the tests reveal about the tissue's response?
Indeed, a key finding, though perhaps not surprising to biologists, was that all 4 types of tissue blocks, whether from the atrium or ventricle, demonstrated a distinctly nonlinear load displacement behavior.
Nonlinear, meaning it doesn't compress uniformly.
Wow. Exactly. It means the tissue doesn't just stretch or compress proportionally to the applied force, like a simple spring might. Instead, its stiffness changes as you compress it. Our sources explain that this nonlinearity is primarily attributed to the complex structure of its extracellular matrix, the scaffold between the cells, specifically the elastin and collagen fibers within it. As you apply load, these fibers progressively unfold, straighten out, and stretch. This causes the tissue's overall stiffness to increase as more force is applied. It gets harder to push the further you push it. Physiologically, the stiffening mechanism is actually vital. It acts as a protective measure for the delicate, smooth heart muscle cells during the heart's natural, powerful expansion and contraction cycles, preventing damage during its constant work.
So the sensor has to accurately measure force against something that changes how much it pushes back.
Precisely. It makes the sensor's job more complex, but also highlights why accurate force measurement is more informative than just measuring displacement or position. The force tells you the actual interaction, regardless of the tissue's changing stiffness. This nonlinear behavior makes the sensor even more valuable.
That makes sense. And drilling down further into the tissue characteristics, they found that ventricles were consistently stiffer than atriums. That seems intuitive, right? Likely because ventricles, particularly the left ventricle, which pumps blood to the whole body, are the real workhorses of the heart. They have a higher fiber content, more muscle, and need to be structurally more robust.
Exactly. They do more heavy lifting, generate higher pressures, so they need to be tougher. It highlights the complex biological environment the sensor has to navigate and accurately report on. The force needed to achieve good contact might differ slightly between the atrium and ventricle.
Okay, so the sensor works on real tissue, and we learned about the tissue itself. But how did they validate the sensor's readings in this biological context? How did they know the numbers it reported were actually correct?
Crucially, the force displacement data obtained from their fabricated MEMS sensor during these porcine tissue tests was directly compared to data collected using a commercial load cell serving as a calibrated, trusted gold standard, indenting the tissue with the exact same 3D printed catheter tip. This direct side-by-side comparison is an absolutely vital step in validating any new medical measurement device. You need to show it performs comparably to established methods. To make the comparison objective, they used the Kolmogorov-Smirnov KS statistical test. This test compares the distributions of two data sets to see if they are likely drawn from the same underlying population. Conducted at a standard 0.05 significance level, the KS test statistically confirmed that the data obtained from both sources, the newly fabricated MEMS sensor and the established commercial load cell, was similar.
So when the KS test confirms similarity between your innovative sensor and a commercial gold standard, what's the practical implication of that for a doctor considering this technology down the line? What does similar really mean in the context of patient care and surgical confidence?
That's a critical question. For a clinician, similar in this context, confirmed by a rigorous statistical test like KS, is a very strong validation point. It's not just a vague resemblance. It means that the new MEMS sensor is performing with essentially the same level of accuracy and reliability as a well-established, commercially available, high-precision instrument, even when tested in a complex, nonlinear, realistic biological environment like heart tissue. This is a major hurdle for any new medical technology to overcome. It tells us the sensor isn't just good in theory or on a clean, simple test bench. It performs reliably where it truly matters. This instills confidence that the force readings it provides will be trustworthy in a future clinical setting. It helps remove that layer of uncertainty, allowing the surgeon to rely on objective, quantitative data rather than just subjective tactile estimation or indirect visual cues. Ultimately, this enhances patient safety and should improve the consistency and success rate of the ablation procedure. It's a foundational step, providing the evidence needed to justify moving towards human clinical trials. #tag.
So let's wrap this up. What have we learned from this incredible deep dive into the microscopic world of medical innovation? We've explored a meticulously designed, microfabricated, ring-shaped force sensor. It can be seamlessly integrated into cardiac ablation catheters. And this isn't just a theoretical concept. It's a tangible advancement addressing a critical, potentially life-threatening need in modern cardiac care.
Precisely. This innovation directly addresses that profound need for real-time contact force measurement during cardiac ablation, a procedure that, as we've seen, has been historically fraught with challenges due to the lack of precise tactile feedback. The sensor offers remarkable performance characteristics: excellent linearity, minimal hysteresis, high sensitivity, and it proved its mettle not just in simulations and lab tests, but crucially on actual porcine heart tissue, performing similarly to a gold standard. This technology truly promises to bring unprecedented precision to a procedure where fractions of a Newton can, as we've discussed, mean the significant difference between a successful, lasting treatment and recurrence, or even severe, life-threatening complications. It has the potential to fundamentally transform patient outcomes by making these complex surgeries more predictable and safer.
And the research paper itself, as is often the case with good science, points to the exciting next steps for this groundbreaking work. It's clear this is just the beginning for this technology, what's on the horizon.
Indeed. While this deep dive primarily focused on the measurement of normal force, that essential push against the tissue, the sensor's clever ring-shaped design, with its multiple piezoresistors strategically placed on each bridge, actually enables the future measurement of shear force components.
Those sideways forces we mentioned.
Exactly. Those tangential forces, the forces acting sideways or along the surface, these are incredibly important for understanding catheter stability and unintentional movement during complex maneuvers inside the heart. Measuring shear force is a key area for future exploration because understanding these multi-axis forces could provide even more comprehensive feedback during catheter navigation and ablation. It could allow surgeons to detect not just how hard they're pressing, but also if the catheter tip is sliding or twisting unintentionally on the tissue surface, which could lead to misplaced lesions. Also, the natural and absolutely necessary progression from here is to move from testing on excised porcine heart tissue samples to testing on human heart tissue samples, perhaps obtained ethically during other cardiac procedures or from tissue banks. This will provide even more clinically relevant performance data, confirming its accuracy in the target species, and pushing this technology one step closer to actual patient use, and ultimately, hopefully, successful human clinical trials.
Okay, so here's something for you, our listeners, to mull over after this deep dive. Imagine a future where every medical procedure, no matter how delicate or complex thing of brain surgery, targeted cancer biopsies, nerve repair, is guided by this kind of real-time, microscale sensory feedback. This could make surgery not just safer and more effective, but potentially profoundly personalized for each patient's unique anatomy and tissue characteristics. What other areas of medicine, or even fields beyond medicine, could be completely revolutionized by sensors so small, yet so incredibly impactful? Think about it, the possibilities are truly astounding, and we've likely only just scratched the surface of what this kind of precision microengineering could mean for the future of human health and technology.